Good evening, everyone. This is Roberta, and I've been curating the biology part for Mid Science for about 2 years now. Tonight is a special night for Mid Science for a couple of reasons. The first is that we have an international guest tonight. So, this is basically my first live in English, so be nice. But also, the second reason is that I finally can host one of my incredible lab mates. So we all have different backgrounds, but we all share the same love for science. Um, Small disclaimer, guys. If you have questions in general, you can ask them using our chat. And if you want to ask the questions in Italian, you are free to do it, and then I will be more than happy to translate them in English to our guest. My guest tonight is Natasha Shylo. She works with an incredible animal model in research. These are incredible reptiles, and this is the veiled chameleon. The veiled chameleon is indeed the real protagonist of tonight, and we will talk about the a lot of things about this crazy animal. Hi, Natasha.
Hi,
How are you?
thank you for having me.
Sure. Finally, I can have one of my lab mates here. So, just like I have usually these first questions that are always the same. For example, I usually like to talk about the career steps that bring you that brought you to your current position right now. So, tell us a little bit about your career.
So I'm going to try to be as brief as possible, but
Yes.
so my I became very, very interested in biology actually in middle school. So I was still living in Belarus, which was where I grew up, and we had a new biology teacher, and she invited me to do a science fair project with her, and it was on ecology, and I really was hooked. I had a lot of fun uh, evaluating kind of environmental aspects and noise pollution in our city. And then my family moved to the United States, and I loved participating in science fairs in high schools here. And then I had a fantastic opportunity to be a summer student at a place called the Jackson Laboratory. They're well known for their mouse facilities, but they also have
Very famous.
a great science program in the summer for which accepts both college and high school students. So that's how I got in. And then that's the point where I realized that I really need to go very general biology
Mhm.
and really need to learn all of biology in my head at that point before I can specialize into ecology or environmental science. And ultimately, I ended up doing molecular biology. And I became interested right around that time. I became interested in developmental biology, kind of
Mhm.
understanding how embryos develop, because so many pathways feed into all the other things, a lot of diseases like cancer, aging, it's all in the small embryo. So when I was
So your major is molecular biology.
so by the time so and what also Jackson Lab was really well known with all their mouse stuff is genetics. So I actually ended
Mm,
up majoring in genetics at
okay.
Cornell University.
Yeah.
And the laboratory, and I worked in the laboratory in my spare time, so to
Yeah.
speak. And my project was kind of fairly heavily genetics, but it was also definitely a
Mm,
developmental biology project.
yes.
Yeah.
I need to interrupt you a second
Mhm.
because this is a big difference
Mhm.
between the US system and the European system.
Mhm.
I would say Italian. I'm not
Yeah.
100% sure for other countries, but basically we start a course that is, for example, my degree is in biology. We don't have other subjects and other disciplines. While
Yeah.
in US, you can select a number. From my understanding, you can select a number of courses and then you can major, so you can
Yeah.
kind of specialize in
That's
the
true.
subject that you like the most, right?
Yeah. Yes. And I have, since we're clarifying things, so technically my degree is in biology, but we had something known as concentration. So my degree is in
Okay.
biology, with concentration in genetics and development. It was one of the less popular degrees for some reason. I don't know. I think it's fabulous.
Who cares? Yes.
And then so as I was when I was finishing up my degree, I mean, I absolutely knew I wanted to go to graduate school, become a professor one day, but I just was not ready to apply to graduate schools. And at that point, I wasn't quite sure what to do with myself. I didn't have the bandwidth to apply that year. So I literally one night sat down and said, well, I've heard there are these postdoc positions once you got a doctorate degree. Is there something after a bachelor's degree? So I literally Googled post-bac position, which wasn't a thing. It wasn't a very widespread thing at that point. It was very new, but it turned out I had one at the NIH, the U.S. National Institute of Health. So I ended up being a post-bac there for a year. And then from there on, I applied to graduate school. I ended up going to Yale University, and that my PhD is in fact in genetics. At that point, where I worked with mice, and I worked with a mouse mutant that has defects in cilia. Cilium is this cellular organelle. It works like an antenna, and it receives and processes a lot of different signals. And it's very interesting because a lot of mouse mutant human patients with mutations that affect cilia have left-right patterning defects. So the heart, for example, wouldn't be positioned on the left side, but it might be in the center, or it might be on the left side. And heart formation is really critical for our life. So a lot of times these patients unfortunately die very early in the first year of life because of the improper heart formation. So I became really interested in left-right patterning, but also the mouse mutant I studied had some very interesting craniofacial defects, and we found out that it has some neural crest cell defects. So what I wanted to do for my postdoc, so after I received a PhD, was to study kind of craniofacial development in neural crest cells, and I wanted to see how cilia affect neural crest cells.
Yes, so we have to specify that neural crest cells give rise to most of the face and the head, so that's why there's a connection here.
Exactly. And so that's how I ended up in my current lab with Paul Trainer at Stowers Institute for Medical Research. And I genuinely came here to study crest cells and craniofacial development. The lab typically works with mice. We have some zebrafish projects as well. The less known project is we turned out to have some veiled chameleons in the lab. And I don't know if it would be possible to bring up a picture now, but veiled chameleons have this very interesting casque on their heads. And we think that neural crest cells have evolved to create this casque. And it's still kind of a mysterious project. We still don't know if neural crest cells are involved in that. Are we going to see a picture?
I hope so. Pandri, can you pull out the picture? Thanks. Okay, in the meanwhile, we can still talk.
Yeah, so they have this casque, and we think it's neural crest cell based. So that kind of got involved in that project a little bit. But what coming from a left-right patterning background, what I knew is that we actually know very little about reptile development. And from my perspective, I knew that we knew nothing about how lizards decide between left and right sides of our body. And it's not just like, up is here, down is there, front and back, and that's how you set up left and right. Well, we actually have a lot of genetics and molecular pathways that dictate how organs go on which side. So I asked my supervisor to work on that project a little bit, and then I had some cool results, and kind of the rest is history. This is what my primary project now. I'm trying to understand left-right patterning, but also a lot of other early developmental processes using chameleons. And they're proving to be fantastic.
Yeah, before going in the science, let's say, but let's talk in general about these animals.
Yeah.
So they have, I think, they have some special features that distinguish them from reptiles, but also in general from all the rest of the animals, right? So can we talk a little bit about that?
Yeah, absolutely. So, so a lot of people, including my husband, will, always disagree with me, but it is a fact that birds are reptiles. So chicken is, for many years, has been our only model organism for all of reptiles.
Yes.
So there is about 12,000 birds. There's about 10,000 lizards and snake species. So, let's say with a couple turtles thrown in there, there's about 25,000 species of reptiles, and chicken was representative of all of them, which is not, of course, there's,
25,000, that is way more than mammals, right?
Yes, there's about 6,400, so 6,400 species of mammals. So there's so many more species, and they're so diverse. Think about a snake and a chicken and a turtle, right? They're all so different. And we think that early development is kind of what sets up those differences, right?
Yeah.
That's and then for many years it was just chicken. So why didn't we have a good reptile species? Well, it's very interesting because there's a number of kind of problems with that. A lot of them lays eggs only seasonally, so you can only get eggs, say, in June, and that's it.
Yeah. That's a problem for research because we need more to study these kind of processes, right?
We need a kind of more continuous supply. Yes. Some of the current animals that we do use, they lay one egg at a time. That's also not very convenient. And what's really kind of more complicated probably for people who study early development is when an egg is laid, the embryo inside of egg, it develops part-time inside the mom, and then it finishes off its development in an egg, on the outside. So that if you were to crack a chicken egg that comes from a farm, and if you looked really, really closely, you might see a little ring, and that's a chicken embryo. So when chicken lays the egg, the embryo is very, very young. But if you crack most reptilian eggs and you open it up, the embryo will have a head, it will have like limbs, it will have a tail. So we're kind of too late to study a lot of things we're interested in. So that's where veiled chameleons come in.
Yeah.
So we think they're very special because when they lay their eggs, the embryo is more like chicken. It's just a little dot there. The embryo is very, very young. It hasn't really started. It's just 2 cell layers deep. So it hasn't started to make a head. It hasn't started making left or right sides or, anything really.
How similar are they to chicken eggs, like externally, dimension-wise?
Oh, I was hoping to bring an egg and I forgot, but the eggs are about
I mean, you can go if you want. I'm kidding. That's
all right. They're about a small, maybe a little bit more similar to a quail egg in size. They're soft. They have this kind of leathery texture to them, kind of like some turtle eggs or crocodile eggs. Occasionally, I will drop one accidentally, and it will just bounce on the floor like a ping pong ball. So they're
I didn't know that.
Yeah, that happens. Yeah, so they're under a lot of pressure. And so once you pierce them, they can sometimes explode. So they're the shell is very, very different. It's not a hard shell. Yeah, so they're kind of small and that's what we had. They're mostly yolk.
Yeah. And you were saying that they are very young when they are already
and then the embryo
mom, right?
is very young when the embryo egg is laid. So they lay the eggs when the embryo is very young. They also lay a lot of eggs. We get 50 to 100 eggs from a single female per clutch. And they also breed, they can breed year-round. And so we get eggs year-round from veiled chameleons. So
we got a coffee. Thank you. Coffee for me and you.
Oh, thank you.
It's a donation, a small donation.
Oh, thank you. Thank you. And so... I lost my train of thought.
Sorry. So you were talking about the
we get a lot of eggs, and from the number of animals, we get about 2,000 eggs per year. So it's a lot of ability to do this. But at the same time, we also our females. They live very happy, healthy lives, and unlike in the wild, the females here in our colony get to live for 3 to 5 years, so they actually can live a pretty long life too.
So how often can they lay eggs?
They lay eggs about 4 to 5 times a year. So what we do is we breed different females in a staggered manner. So it's not always the same female who's laying the eggs. I mean, they do, but we just have them staggered. So we have a continuous supply of eggs year round, but it is, it's not, we're not frustrating the same animal. Yep.
So, so far we have talked about these great animals, the little chameleons, and we are just like talking about their special features, what make them a little bit different from other reptiles and other animals in general. So we talked about the early developmental stage when you open an egg. So when the egg is outside the mom, we talked about the fact that they are very prolific. So you can have, you said from 22
50
to it's a to 50 to more plus eggs. But what else? What does distinguish chameleons?
Well, and then of course, so now we have a non-avian model of development, right? What we found is that, and this is going to get a little bit technical, but so most of the time when we study early embryos, a lot of, the preferred mammal to use is mouse. But mouse embryo shape is actually very weird and very different in the early stages. I'm not going to usually show this for real, but what I'm going to tell you is that mouse is kind of bent backwards. So its heels, so the heel of its foot is kind of almost touching its head. So it's kind of this U-shaped embryo. And that is not how a human embryo is.
No.
A human embryo is kind of like a little dome, like this. And that is kind of, there's say, and then it's flat on it, and it's on its back, I believe. And then whereas if we look at the chicken embryo, up till now has been the most similar embryo we've had to study that, but the chicken embryo is completely flat. What we found, which is absolutely fascinating and was very unexpected, is that chameleon embryos is actually a whole lot more similar to human embryos than pretty much any other model we've had so far. They also have this like little little domey shape too, that is kind of quite reminiscent of what a human embryo looks like to me. So we're very excited about having a model organism to be able to study those really, really early stages of development, and we think the genes.
And this is very this is very surprising, because usually if you take, for example, a developmental class, what we are usually taught is actually that until a certain point, all the embryos look the same. But so this is not really true, like not totally true.
Well, actually, not. Yes, it's not exactly. I think the clock, the developmental clock you're thinking about, it's more of an hourglass. And there is a stage where all the embryos really do look very similar. If you've ever seen
We're talking about different species, yes.
Different species of animals, yes. So if you see, look, if you line up a picture of a human embryo, mouse embryo, a chicken embryo, if zebrafish embryo, they have this kind of the head, they have a tail, they have, things are kind of folded up on that. So that sort of happens in the center. And then after that, all of a sudden chickens start getting feathers and, things like that. So they all get very different. But early on, what we've also learned is that at some very, very early stages, the embryo is also quite different. And we don't necessarily have all the models to study that.
Exactly. So, but what about like the adults? Talking about adults, right? Chameleons, I think, are well known for their mimetism. And also they have some other anatomical features, right, that are very special.
Yes. So, so we've talked a little bit about their casque. We think it is neural crest based, but we don't really know. We haven't really answered that question yet. We've looked a little bit in the neural crest cell, kind of migration, formation, and face formation in them, but that project remains available. What some other cool, everybody knows about the color change. I don't really work on color change, but what I know is that what is thought is that within the cells, and I'm going to butcher this a little bit, but there are more or less like crystals that sort of they're able to sort of change all of them, and it's a fraction type of system that changes the color of their skin. so it's not like a different pigment. It's more about the
Mhm.
way it reflects light. So that's a well-known chameleon feature. Some people do study their projectile tongues because they can
Oh yes,
they hunt
yeah. We had
quite
we posted a video on Instagram yesterday about that, and it's
exactly.
very cool.
Yeah, so that's
Because they can also grab things with their tongue.
Yep,
So it's not only the length that changes, but also they
exactly.
have muscles to grab things.
And it's incredibly fast. I've seen it in action. It's
Yeah.
just like
Oh, did you see it?
yes. So you know, as I said, people do work on it, including veiled chameleons. What we have been interested in is that their limbs are modified for their arboreal lifestyle, so they live in the trees. So instead of having, so they still have 5 digits, but their digits are actually fused, so it's a little bit more like this. And it's I always forget, but it's actually different between their what would be their hands and their feet. So one of them is more like this, and the other one is more like that. But what it
Okay.
allows them is to grab onto branches really nicely. For us, it would be kind of like this. For them, it would be, 2 digits on one side, 3 on the other. So they can really, really hold on much nicer to the branches they're on. And also their tail actually works as a fifth arm. So they're able to twist it, around those branches or your hand if you're holding them, and they hold on. They help themselves to hold on to it things. So we have done a little bit of work in our lab looking at the limb patterning and how we could molecularly explain this. So what happens is in the human development, right? So we all, as during that stage we talked about in the hourglass, at that stage, all embryos, when they limb, you look at their hand, it's just a little bud. And then eventually you get condensations to what will be digits. And then what happens is known as programmed cell death. So the cells will die, and then you'll have individual digits.
Yes.
Right?
Apoptosis.
So apoptosis. So in humans, the condition of when there's no cell death, so then say there's digits that are fused, known as syndactyly. So in chameleons, it's actually effectively a programmed
Physiological,
syndactyly
yes.
condition. Exactly. So we're very interested to find out, why is it that between these digits, 3 and 4, cell death happens, and it doesn't happen between digits 1 and 2 and 3, right? And kind of what are the mechanisms for that? And also, how can we translate what we understand from that into human patients with this condition? So there's a lot
potentially also a model for studying human diseases, right?
Exactly. Yes. That's always being able to study human disease was always in the back of. Yeah. Yes, that's kind of some of the projects we've worked on in the lab, and I've been involved with. Yeah.
But you also studied left-right pattern,
oh yes,
right?
of course.
Yes.
So that's what brought me
You missed
to animals.
the juiciest part.
Exactly. I get distracted by all the other fascinating features, but that's sort of what brought me here. What's very interesting is that, so I told you that I worked on cilia a long time ago.
Yes.
And the reason why we're interested in, the connection between cilia and left-right patterning.
Exactly.
It was established in 1970s, and there were people with a condition known as, at that time it was known as Cartaginer syndrome. Now we know it as primary ciliary dyskinesia. So what happens is those people would have frequent bronchitis. So they would, bad lung function. There would have male sterility. And one of the other features that you would often see them is that the organs will be a mirror image. So their heart will be on the right side, and their stomach will be on the right side, the liver will be on the right. And some of it made sense because, say, male sterility. So a sperm tail is actually a cilium. So cilium, and then in our lungs we have cilia that beat and remove mucus. And so when researchers had looked at the structure of those cilia, they realized in those beat. So they can't remove the mucins. They can't get to the sperm can't get to the egg. So it seemed like this was a disease of non-motile cilia. So the cilia are no longer motile, and that's what's causing these phenotypes. However, what the connection was to left-right asymmetry was really strange. And so since then, we've learned a lot. So turns out there is an organ in mice and humans that is known as in mice it's known as the ventral node. It's a transient organ.
We are talking about something that happens at early developmental stages when the embryo still is not really patterned, so they need something to guide the cells to the right position in the body.
Exactly. So that happens very early. It has this teardrop shape in mouths. It's kind of like a little ditch, and it turns out that it has cilia in it, and those cilia beat and then create a flow of liquid. We don't know all the cell mechanism, but we know there is some sort of motion from right side to left side, and somehow that fluid flow made by motile cilia gets translated in asymmetric gene expression only on the left side and not on the right side.
That's
what we know how happens. This is what happens in mice. We think that's what happens in humans. And it seems like that is how what happens in most things that are left and right side. So we know that it happens in it's a similar mechanism. So motile cilia are involved in zebrafish, in frogs, in a chordate siona. It's involved. So this motile cilia is very ancestral.
A very early chordate. Yes.
Yeah.
It's actually thought to be, I think, one of our sister group, like to humans, right? So Siona is a very small animal that has the notochord, that is the ancestral structure that then gives rise to the, what's it called? The
spinal cord eventually in us. Yeah.
Spinal cord, yes.
Yeah. So we think having motile cilia is a pretty deep evolutionary feature, but we knew that in chickens their node does not have motile cilia. So
how does that happen?
We don't know. So somehow they established left and right asymmetry using some other different mechanism. We think there is some sort of asymmetric cellular migrations. Now, so that's chickens. And that has been shown in other avians. What's weird is that pigs also don't have motile cilia in their things. And it seems like that group, so they're even-toed ungulates. So think pig, whales apparently are closely related to pigs, cows, things that have kind of, toes that go like this, like with a double, not a horse. Horse just has one toe in the center. The things that have double toe. So those, all of that branch of animals, for some reason, don't have cilia in there, a motile cilia that help with establishing left-right asymmetry. So I said chickens don't. So when I came in the lab, the question that started me working with chameleons was, well, how about other reptiles?
Exactly.
And not avians. So what I found.
So did you find cilia in the chameleon embryo?
So okay, I'm gonna be careful about answering this question because yes, there are cilia, because cilia are present on pretty much all cells in most organisms, but there's no motile cilia. So chameleons have motile cilia at later stages of development, but not at the time when they establish left and right sides. Somehow, similar to chickens, they don't use motile cilia. I see some big rearrangements in their body, but it's not motile cilia at that point.
We have a question from the audience. That is perfect because it says, if a common ancestor has a property, so I think it's a feature or something like that, like motile cilia, it means that chicken has lost this property, but seems quite important to lose. Like, I think that is very important to lose. What do you think?
Yeah. So I think that's a question that has drawn me in and like positioned me here forever. I think I would love to learn the answer to that question. But we do think that this property has been lost, and it has been lost twice. So it has lost in all of reptiles we know now, and it has been lost in those even-toed ungulates.
Mhm.
And it does seem like they both do use somewhat similar mechanism. There seems to be some sort of asymmetrical cell migrations that somehow drive the asymmetric gene expression. So
Mhm.
so what we think could be happening, because sure, I told you that Siona, I did mention sea urchins also use motile. So like sea urchins, right? All of
Mhm.
those animals use motile cilia, but fruit flies have left-right asymmetry.
That's
Snails
true.
are left-right asymmetric. So there are other mechanisms to establish left-right asymmetry,
Mhm.
something that has even deeper, more evolutionary relatives. So what we think, for whatever reason, there seems to be some sort of reversal to a more deeper ancestral state. And
Hmm.
but because we have been so focused on mice and zebrafish,
Yes.
fish, we've been very tunnel vision focused on motile cilia and their role in left-right asymmetry. So we know a lot less about those other mechanisms of
So
establishing.
we think the motile cilia system is the first or is the most important, but actually there are others, right?
Sort of,
I
yeah,
know.
I would say that. Yeah, I think motile cilia are still incredibly important, but I
Yes.
think there's more to it. There might be
Yes.
mechanisms that we're missing because we've never looked for them. We didn't
Yes.
know to look for them.
Mhm.
Yeah, and what's
So going back to chameleons,
mhm,
so what did you find? Like we were
yeah,
talking about that, and then we
yeah.
changed
Well,
direction a
so
little bit.
I mean, so in a nutshell, we found that there is no motile cilia at the time when left-right is established.
Mhm.
What also is interesting, we see that the embryo, so in the chicken, there is a very well distinct node and the cells migrate around it. I don't quite see that in chameleons. What instead I see is the body does this whole body wiggle, kind of this, and then
I saw the videos. They are very cool.
somehow
It
after
does make.
that we have a left and the right side. And
How do cells know where to go?
I have no idea. So I no, I have no idea. If we do really start kind of be more philosophical about it, and
Yes.
really do go back into some of those early things. So what we think happens in Drosophila, so fruit fly,
Mhm.
we think that there is, if we kind of go deep, smaller and smaller and smaller at a molecular level. So there are, for those who study chemistry, in organic chemistry, for example, there's molecules, right? How atoms are attached to each other. They can be
Mhm.
attached like this way or this way, right? So
Yes.
there's molecules have sidedness to them.
Yes.
So actin is a macromolecule in our cells that help kind of drive, say, migration of cells.
Mhm.
And actin is apparently a sided molecule. So we think somehow it's possible that things like actin, for example, so as it
Can
gets built
drive.
on, it
Okay.
can sort of mechanically, because of its inherent asymmetry of that macromolecule,
Mhm.
it can kind of drive the migration of cells in a specific direction. And then some really nice papers out there
That's
on this
insane.
small level. If you look at cells growing in a dish, so it's
Mhm.
a single layer of cells, and if you really watch them, after some time, I think it's not all types of cells, but some types of cells will just start circling in circles around the dish. So they will just crawl around, I think, counterclockwise.
Yeah.
Um,
Oh my
and then
God.
just
Which
kind of waiting that
cells?
happens.
Which cells?
Oh, I
I
can't
don't
tell you off
remember.
the top of
Okay.
my head,
So
but
the question now is what drives the change, right, in the actin, right? What is the signal?
well, so actin is just built asymmetrically, right? And it is
Okay.
whatever the mechanism that kind of drives the buildup of actin to just
Mhm.
for a cell to migrate. But that will be a cell migration question, and I
Yes.
guess I'm in a good
Anyway,
lab
we are
for
going off
that
topic.
because
No, go back. Let's go back to chameleons. um,
yeah.
so, for example, if a student was to say, okay, I fell in love with chameleons,
Mhm.
what else could I study, for example? If I don't know, is it possible to work with adults, like
Um,
bureaucracy wise?
so so it depends, right? So we hmm. So I'm very I guess insulated in kind of
Mhm.
where I'm at. We use them as a model to study early development. As far as I know, here at Stowers, we have the biggest research colony of veiled chameleons for those purposes. And most of the people I kind of interact with, we all study them for the purposes of kind of early development. But veiled chameleons we talked about why they're great for lab, because they breed a lot, they make clutches of eggs, but they're also, they're popular pet because they're relatively easy to take care of. Well, that also makes them a good invasive species. So in the United States, chameleons are actually invasive species. They're present in both Hawaii and Florida. And I know that parts of kind of, I think maybe Southern Europe and Northern Africa also have areas where veiled chameleons are invasive. And I'm not as familiar about it, but I think there are, people working with that. And in that case, it's a very different perspective.
They are regulated.
You would work with them. But, people have looked at them from those perspective, kind of distribution of the invasiveness and things like that. One other
I have a question.
Yeah.
Okay, no, go ahead.
So one other thing is what we are interested in doing is that, and that touches both on conservation of, or on kind of population management, but also because they're so populous, they're good. They'd be great for us to use as a model to see if we can figure out how to do IVF, so in vitro fertilization in reptiles.
That's great.
So because we can work with a species that is, has a lot of eggs, we have a lot of access to adults. We can collect sperm from males and females, and then we can use what we learn in chameleons for conservation of other reptiles, which are are endangered, for example.
That's true.
So that would be kind of work that would happen with more adult animals.
So I don't know if we can share this information, but you mentioned that Stowers, that is the institution where we work at, has the biggest facility, right? So how many animals do we have here? I don't know if we can share this though.
I think probably okay. I think we have we have about 20 animals typically. They, and as I said, they live really happy lives of 3 to 5 years, and they're quite spoiled here, I have to say.
Most of them have a name, right? Names. They have names.
Each one gets a name. They have very they have big personalities. And each one of them get a lot of very special attention here.
Do you have any fun facts about chameleons in the facility, for example? Something about a parade or something?
I know what you're. So my friends here, I mentioned it and they find it absolutely fascinating. But we all know about chameleons and their changing colors. Well, it's actually a bit of a misconception that they change color to blend in with their surroundings. Maybe a little bit, but they actually use color changes as a way of communication. So typically a happy chameleon is going to be this nice and green color. But when they're angry, they kind of turn, or when they're upset, they turn this kind of brown color. But when they're angry and they try to be threatening, they turn this like dark blue, almost black color. So what happens is I said that we breed them year round. So we would, what would happen is we first need to know, they're territorial. So we need to know if a female would want to breed with a male first. So what we do is we parade a male in front of female cages. And if a female turns, they just go very vibrant, very bright colors. So she's a happy colors, then the male visitor is going to come and visit her. And if she turns this black blue color, maybe starts hissing, then we know that she is not having it today. And then the male just kind of will keep him going off to another location.
that is so funny. And they also have quite big cages, right? I remember I saw the facility once, and it's very great the way they live.
Yeah, they
Yes.
have space to climb. They have a green leaves to
Hmm.
hide. They have a lamp that produce. So with chameleons, it's very important. They need UV to produce enough vitamin D for good kind of bone structure and proper calcium absorption. So we have special UV lamps and heat lamps, so they can choose how warm or how cool they want to be, and we kind of closely monitor their health overall, including their bone health. They
What
get
do they eat?
they eat so in the wild, veiled chameleons will typically eat mostly insects. I've heard them eating actually small lizards sometimes too. So
Oh,
they're carnivores really. In our facility, they're more omnivores. So they typically
yeah.
eat they eat crickets, which usually we just kind of shake some supplements on crickets, like calcium, for example. Uh,
Hmm.
but if we just feed them crickets, they get tired of them, and they get kind of picky and they stop eating. So in addition to crickets, we always make sure they have some bright vegetables. So they get
Hmm.
peppers, they like sweet potatoes, pumpkin, um,
Wow.
but yeah, carrots.
That's a great diet.
Yeah, and then but they also we also give them some other things like mealworms, hornworms, super worms.
Wow.
We breed two species of cockroaches as a way to feed them as well, so that there's kind of a good variety of diet. But also, it's interesting to think about it because some of those insects we do breed, and what you feed. Kind of what we feed those insects is what our chameleons will ultimately eat. So we also want to make
That's
sure
great.
that the insects we
Yeah.
have in our colony eat
They are healthy.
good,
Yes.
healthy food. Exactly. So they get
I
good vegetables.
remember last year you telling me that they also prefer colorful veggies. Am I
Yes.
wrong?
Yes, and we don't know. I don't know as much about their. So we know that chameleons see in color, but I don't know as much about their
Yeah.
overall. But they do seem to prefer things that are colorful, like peppers, which would be yellow or orange or red. We tried like lettuce that is green, and they just
Mm.
completely ignore it.
It actually make also sense with the fact they communicate with colors as well,
Ex-
right? So they
Yeah,
have like these vivid colors when they are happy, and they are just like, no, I don't want colors. I don't need colors because I'm colorless right
yeah.
now when they don't want to communicate, right? So that makes sense. And they also have a particular feature about the eyes, so they can move
Oh,
them, I
that's
think,
right. How did I
independently, right?
forget that?
Yes.
Yes. So the eyes in chameleons are independently movable.
Mhm.
Yeah, so I have no idea. The brain must be fascinating how they can process
Yes.
all of that. But I think it's also really funny when you come in and you're past a cage of the chameleon who, is the face is directed in one way, but you could tell that they are looking
Look,
at you because their eyes completely rotated to the back and they're
so
just.
they go like 180, right? They
Yep,
have
they can go 180,
180.
or one of them could be looking over there, where the other one will be looking back or front. It's very disorienting.
And it's also crazy because the eye is quite big itself, but
Mhm.
then the part that is exposed is quite small, right?
Yeah, yeah.
Why? Why
I have
is
no
that?
idea. No, I don't know that. Yeah, I don't know. And I wonder if it's a little bit of a reptile feature because if you look at chicken embryos, during development, their eyes are also enormous.
Hmm.
And I wonder if it's one
Oh,
of the
yeah, that's true.
things that the eye sort of stays a similar size as the animal grows. I'm not going to speculate because I don't know. Yeah.
So we have another question. This is a funny one as well. Are they sociable with each other and with humans, or they prefer to be alone?
They're not particularly social. They are fairly territorial. What we have to do is we need to, um, we have kind of barriers between cages a little bit, because we found that if we have two cages with two males facing each other, they will kind of display and threaten
Wow.
each other from their cages all the time, and they will do it at the expense of like eating and sleeping or whatever. So we found them to be like start getting like unhealthy when they do that. And then also if you have them facing a female, they will display so that she can come to them, which also takes a lot of their energy. It's not particularly healthy for them either. So we give them breaks, and they usually have a little barrier for at least a couple days, so that they can like not, but it's also interesting if they are always kind of, if we were to keep them in a box, for example, which would never do, they don't really see other chameleons, then when we do introduce them to chameleons, then they end up being quite aggressive. So we have to do this fine balance of they still see that there's other chameleons in the surroundings, but they don't see them at all times, so they don't have the need to protect their territory at all times. So they're not terribly social, I would say that.
Yes. And also I would say also with humans, and since they are and but I think since they are research animals, I don't think you can go and just pet them, right?
Not really. We do find that for some projects we have to handle them a little more than others, and we found that at first they were like, wow, why are you taking me out again? Like, and then these days they're like, okay, it's like that thing again, all right. And they also recognize people, but when the veterinarian comes in to check on them every day, they just all like hide extra well so she can't.
Really?
Yeah.
Wow.
How they know that she's the one who would poke and prod them.
Yeah, because we also have this protocol, so a veterinarian gets in and just checks that all the animals are in good health and they are treated well. So I would say they are very spoiled.
Yeah, they have good lives.
How many labs roughly work with chameleons? Not only in US, I would say also like Europe, for example, because I think we have people mostly from Europe.
Yeah, our lab here. So the graduate student who started Veiled Chameleons and worked the Veiled Chameleon project in our lab, he now has his own lab and he does some Veiled Chameleon work. I'm aware of one lab in Czech Republic who sometimes works with chameleons. They actually study craniofacial development, and they've looked at things like palate development in chameleons. So palate is the top roof of your mouth, so they've looked at that. And as far as development, as far as more dedicated, that's it. That's that I know of. So we collaborate a lot. So we send fixed samples or sometimes eggs out to collaborators. So we have a couple more labs that we work with directly, but kind of on their own, those are the only three that I really know of. But there's a couple more people who, as I said, study more of a kind of conservation and like more wild, like environmental relationship between chameleons and things like that.
Do you know if there is any other reptile animal model like that is used in research?
Yes. So the most popular one these days, I think, would be a brown anole lizards. I believe they're native to Cuba. In the United States, they're invasive in Florida and the southern United States. So a lot of people work with those. And there are, if you look at paper, so there are some people who work with geckos. Gecko has a very interesting, I think people are very
interested. I love geckos.
For their digits because they can, very sticky, they can crawl vertically up. So people work with geckos, and there's some labs that study a few species of turtles. In particular, I know some of them are particularly interested in sex determination in turtles because that happens through temperature. Exactly. And kind of what is the mechanism of this temperature dependent sex determination?
I read a paper that is actually through intron retention. That's crazy.
Yes. Yeah.
We have another question. So how are the ethical standards and requirements? Are they different from other more common animal models like chicken? Takes a long time to get permission.
I don't know if I would want to comment on details about that because I
Yeah.
think
Okay.
it's different between the United States and Europe. So I don't
Yeah, I think
inform
so.
anyone. Yeah. Yeah.
Okay.
Yeah.
So we were talking about turtles, for example, and then I think that's snakes. Do people use snakes?
So, I mean, they have been used. People have looked at snakes and stuff like that. I mean, obviously people have been interested in like, how did snakes get this long, right? So,
Okay.
the formation of ribs, but I'm not
And
quite
they also,
familiar with
oh,
who does it
you
routinely.
remember me reminded me about the ribs. There's another feature, right?
Oh, that is yes. So we are very
Yeah.
interested in the ribs formation because
Mm-hmm.
so for us, if you poke yourself in the belly, if you're very squishy down there, but in chameleons, the libs actually go all the way down to the pelvis, and we're very curious why, and whether it's to protect the eggs or some other feature about their lifestyle, we don't know. Yeah.
So do they have a long sternus?
The sternum. So I think the true sternum is long, but it doesn't. I don't think the true sternum go all the way down to the pelvis.
Okay.
But the ribs are still connected with some thinner
Cartilage.
cartilage there. Yeah.
Cartilage.
So they're still connected all
Okay.
the way down. Yeah.
That's very nice. Let me think about some other questions because we wrap up because it's almost time. Where do you see like chameleon research in the future?
Well, I'd like to. So I'm hoping to get a faculty position, so be a professor, and have my own lab. I'm interested in working on, kind of developing further this kind of questions about left-right patterning, what the evolutionary kind of background of all of that is. I'm very interested in some of the earlier stages of that to understand, a process named gastrulation, which is the way we go from having 2 cell layers to 3 cell layers. A third cell layers is what would be muscles and, bone sort of stuff. So I'm kind of very interested in that process. That's the process when the embryo looks a lot like human. But I think they're just a really cool organism. And people, whenever I give a talk, people are like, well, how about this question? I'm like, I don't know. We don't know anything about it. I think
Exactly.
there's a lot of room to study the limb patterning and the eyes, whether they can see color, how the ribs are formed. We've recently sequenced their genome, and
I was getting there now.
people interested in the genome, there's a lot of very interesting features. They have these little tiny chromosomes that are not really present in mammals, but they're present in a lot of other animals, and they seem to be just like evolutionarily just mix and match in all kinds of different
How many
manners.
chromosomes do they have?
So they have what's known as so let's put it that way. So in a sperm or an egg, right, which would have kind of just one set of chromosomes, you would have 6 macrochromosomes, so 6 big chromosomes and 6 microchromosomes. So within an individual, you'll have chromosomes from mom and dad, so you'll have 24 chromosomes total. Yeah, and then so 6 of them are pretty big and 6 of them are really teeny tiny, and kind of how the evolution of how that's happening is very interesting. We also are interested, in looking at sex determination in these guys, because I told you turtles are used in temperature sex determination, and it turns out that reptiles, they've reinvented sex determination many, many times. Unlike all mammals that have X and Y, birds have what's known as the ZW system. So unlike humans where XX is female, so let's see, ZZ will be male. Oh, I'm going to get this wrong and then I'm going to regret it. And then ZW will be a female. So a female is the heterogametic sex. And then, but these systems have re-evolved many, many times. So veiled chameleons have an XY system, but it has nothing to do with what mammals have. So kind of they invented their own XY system. And there are other reptiles who've kind of reinvented any of those systems many, many times. And some of the sex determination is just wild in reptiles. So I think having a lot of these genomes will allow us to kind of understand how sex is determined and how it's genetically encoded, and kind of what are the pathways and the processes that feed into all of that.
Let me think. Like, I'm very like curious about these microchromosomes.
Mhm.
So why do you think they need microchromosomes?
I don't know why those microchromosomes exist necessarily, but what we do know is very limited. But they seem to have a lot of genes. So for those, if you guys don't know, that our genome has, in between genes, there's a lot of space with regulatory elements, repetitive elements, stuff that used to be known as junk DNA. So the genes are actually, we used to say like 1% of our genome is actual genes,
Mm-hmm.
right? So it's really not a whole lot of, but on those, if you look at those microchromosomes specifically, there's a lot less of that junk DNA, and there's just a lot more genes. They're a lot more enriched in those. So there seems to be a lot of genetic material, very important stuff, and we don't do them fusing and breaking between different species a lot. We think that some of the genes that are rapidly evolving are present in them because there's those break and separation.
Very close.
One of the examples that in the literature is the snake venom gene. So most of the venom genes are apparently located on those microchromosomes, and apparently venom in different species of snakes, they're all quite different between each other. So those are kind of some hypotheses that are out in the field, but who knows why they're microchromosomes and what
very
they are.
interesting.
So chicken actually has, so like I told you, chameleon has 12 chromosomes, right? As in a mom, I think chicken is like 38. There's a lot of, and they have a lot of those teeny, teeny, tiny chromosomes, and who knows what genes they have on them and what they're doing. Yeah.
So now basically the genome is available. So if I don't know, anyone can
almost. So when we have a discovery, we usually write it up into a paper and then publish it. So we're hoping to submit and publish that paper really, really soon, hopefully this year. And the moment it is published, the genome will be publicly available to everyone. But right now, we're still working and making it a little pretty because it takes time. It took years to assemble human genome. It's faster now
because you did from scratch, right? It was not. Yes.
Yeah.
So I think we are at the end of this amazing live today. I really want to thank our guest. She is very nice. I didn't give her a lot of time before to decide, so she just said, "Yes, let's do it. Let's talk about science." And I really want to thank the audience from tonight. Thank you for following along. And I don't know if Natasha wants to say.
Oh, no, absolutely. Thank you for having me. And I'm on Twitter. You can find my contact information on Paul Trainer's website. Feel free to ask me questions if you're interested. I'll be happy to answer whatever questions you might have.
Okay. Yes, if you have any questions from Natasha, feel free to reach out to Meet Science, and somehow we will make it happen. Thank you, everyone. See you next time.